I just want to make sure we're not looking at fossilized material here, fossilized tissue. This that's been replaced by calcium.
These are the cells.
These are real cells.
Correct.
Well, Kevin, I understand you published all of this work.
Yes,
is that right?
This work has been published. We've actually made the cover of American Laboratory. We also published in a journal called ACTA Histochemica, and that's a more technical article. It goes through and explains then what we did, how we processed the horn, and then of course draws some conclusions from that. Particularly the conclusions are that even though it's a horn, which is different from a bone, it still had tissue. Even though it was wet when we pulled it out of the ground, it had what we call matrix, which is another word for mud. It still had tissue, so just a different specimen than what had been analyzed before. No one had done a horn before. Now we start getting closer, and what you're going to see then is you're going to see little pieces of tissue there, and that's what you're going to pick off and then put under the higher powered microscope. Okay? And then under the higher powered microscope, see, now we look at the tissue itself, and there's the cells.
Oh, it's fascinating.
See, and then there's another. And like I say, those are very specific osteocytes. Now, those osteocytes have a very unique structure. Okay, now we're coming into some of the texture of the horn itself. And see, this would be a blood vessel. And if you look, you see, this is dissolved away. This would be the interior of the horn. And you can see all the detail that'd be left in there after you take the calcium away. And then there, of course, is an osteocyte.
So that has to have shaken up the scientific community. What's been the response of all of this?
The initial response, when Dr. Schweitzer first published her work, which is what became very popularized in 2005, it generated a lot of response that previous papers had not generated. And there's kind of a question of exactly why it generated so much response. But in 2005, her paper was published in the journal Science, which is a very broad distributed journal, very highly respected journal. Color pictures, you can't minimize the impact of color pictures. So you had people that would be more biochemistry and biology backgrounds that maybe for the first time paid attention to this. So that generated, I think, some not only publicity for it, but certainly some controversy. And so initially, some of the reaction was rejection. Oh, it's contamination. That's not really dinosaur. It's microscopic artifact. It's bacteria, because bacteria can look kind of strange sometimes. So you had a lot of proposals of what it could be. And to her credit, Dr. Schweitzer did more work, which is what science is. She did more analytical work, dug deeper. They began to find protein. You break open some of these cells. You look at the matrix these cells are attached to, and they're protein. Particularly one of the common proteins they found is called collagen. Now, collagen is the most common protein in any vertebrate. Vertebrate meaning those animals that have spinal columns. Collagen is the most dominant protein. It's a hardy protein. But there was no reason based on any kind of biochemistry known about collagen, any kind of biochemistry of how collagen degrades. There was no reason to think that collagen could naturally, easily survive for 65, 70 million years.
And all of that research, did it lead towards the conclusion that it's not bacteria, it's not something that
it very much did, right. That you can dismiss the bacteria idea, you can dismiss the contamination idea. It is real dinosaur tissue.